introduce our moderator for today. And it's Dr. Kamal Dua, who's all the way in Australia. And as I said, we really appreciate that he has been able to come with us. And this just shows how important this international flavor that we have on NeuroCovid is. So Dr. Kamal Dua is an award-winning pharmaceutics and drug delivery researcher who has deep roots in teaching, research, and industry spanning 3 nations and 2 continents. He holds dual expertise in pharmaceutical sciences and immunology and microbiology. And he also completed 4 professional diplomas in the areas of clinical research, product development, and drug regulation. Kamal's academic research has contributed significantly to the field of managing chronic inflammatory diseases, especially airways diseases, using novel nanocarriers to deliver potential therapeutic molecules, including phytoconstituents. His research identifying potential key targets, regulating the pathophysiology of the disease, and formulating them into effective drug delivery systems that could be translated into clinical settings. So tremendous to have you with us, Kamal, and I'll allow you to introduce our first speaker, who will be Dr. Leo Galland. Thank you very much for joining us, Kamal.
Thank you, Philip. That's a pleasure. Thank you so much. Hello, welcome everyone to the 4th Long COVID Coalition Congress on NeuroCovid. Valentina has nicely presented already what's the relevance and importance of this coalition congress. And without any delay, I would like to introduce our first speaker, Dr. Leo Galland. Dr. Leo Galland is a pioneer in studying the impact of intestinal microbes, the gut microbiome, and intestinal permeability, which is called leaky gut, on the health and disease. He has received international recognition for developing innovative nutritional therapies to treat autoimmune, inflammatory, allergic, infectious, and even gastrointestinal disorders. He has described his work in numerous scientific articles and textbook chapters. He's a graduate from Harvard University and New York University School of Medicine. Dr. Garland is board certified in internal medicine. He's listed in leading physicians of the world and America's top doctors. In 2017, Dr. Garland was awarded the Albert Nelson Marcus Lifetime Achievement Award by Marcus. Who's Who, Dr. Garland. Who's Who. Dr. Garland is regularly chosen by New York Magazine as one of the best doctors in New York. So without any delay, I would like to invite now Dr. Garland to please share his screen and deliver his talk. Thank you.
In the past 10 days, there have been 4 major papers that have appeared that I've had to add to this presentation because this is such a dynamic area. And I expect that this is really just a work in progress. And it is based on my reading of the literature and the practical steps that I take with patients who have long COVID. There is a roadmap that I attempt to follow to reestablish a healthy gut-brain axis after COVID-19. It's well known that there are disturbances in the gut microbiome associated with COVID-19. Some may predispose to severity of COVID, some predisposed to long COVID, and I'll highlight those, and others are probably the result of having COVID. And I think those are becoming increasingly important. The steps that we will follow are to stop viral proliferation in the gut, reduce epithelial cell inflammation that's in the gut, reverse the gut bacterial dysbiosis that is created by COVID-19, restore the transport and metabolism of tryptophan, which is impaired in people with COVID-19, restore the mucus layer in the GI tract and tight junction integrity to assure a return to normal intestinal permeability, detoxify the intestines, and then I want to create a microbiome that will support T memory cell function, which is essential not only to recovery from neuropath, but also to protection against the effects of future infection. Now, There's abundant evidence that persisting viral infection contributes to long COVID, or PASC, as it's called in the US in scientific circles. I'm not going to review all of these studies. The most recent was a study from Harvard which found circulating spike protein in the blood of people, in the plasma, not even in cells, of people with COVID-19 up to 12 months after they'd first been infected. And these researchers said there has to be a reservoir of chronic viral infection that is present and that is creating this and then driving, presumably driving long COVID. And the immune response of people with neuropasc in particular, this was studied at Northwestern University and is referenced here, shows an impairment of T effector memory cells of a type that would be seen with chronic antigenic stimulation. So steps to prevent viral proliferation in the gut. There are drugs that might be used. I mostly are using natural products, and flavonoids figure very strongly into that pharmaceutical repertoire, especially those containing quercetin, curcumin bicalin from Scutellaria bicalensis, glycyrrhizin from licorice. They have effects of protease inhibition, and they also destroy bacteriophages. And as Valentina will be explaining later, and there was data presented on this at the last conference, there is very strong evidence that the virus, the SARS-CoV-2 virus, not only infects human cells, but can infect bacteria and act as a bacteriophage. Based upon that data and the work of Dr. Carlo Brogna and his colleagues at Cardiomed in Italy, I am now exploring the role of antibiotics along with these antiviral herbs, in particular amoxicillin, which in his studies actually proved to be the most effective. I'm also exploring the use of oregano oil, because oregano oil has both antibacterial and antibacteriophage activity. And in a review of dietary factors that could impact the activity of bacteriophages, polyphenols, especially the bioflavonoids, were identified, and oregano was found to suppress multiple types of prophages, preventing them from incorporating into the bacterial genome. And what bacteriophages do is they are viruses that enter into bacteria as become prophages, then become incorporated into the genome, activated, and they may kill or lyse the bacteria or change the metabolic activity of the bacteria. Of note, the natural sweetener stevia was shown to promote the activity of multiple species of bacteriophages, increasing their impact on bacterial hosts. So since we don't know what stevia does in this particular setting, I would use caution with stevia in patients who've had COVID-19. Now, there also is a probiotic that has antiviral activity. It is Bacillus subtilis B7092. It is a particular strain. If you can find this, when I have access to it, I use it. It was developed in the Soviet Union about 40 to 50 years ago for the purpose of delivering human alpha interferon through a plasmid into the GI tract. And SARS-CoV-2 virus is very sensitive to killing by alpha interferon. It's available in a few countries. I've mentioned the names. It was actually developed in Ukraine initially. So I will use, in the initial stages, I will use these antiviral herbal products that are rich in flavonoids, along with antibiotics, and then follow that with Bacillus subtilis B7092. The next step is to reduce epithelial cell inflammation. the idea that you can just go and correct the dysbiosis of an inflamed intestine with probiotics or prebiotics, I think is incorrect, because inflammation itself alters the milieu. Inflammation increases the synthesis of nitric oxide, which results in an increase in the nitrate concentration of the inflamed tissue. Now, a high nitrate environment, which is what's created, enhances the growth of many bacterial pathogens, but it inhibits the growth of many healthy anti-inflammatory natural bacteria. We see the same thing in streams and in water when high nitrate detergents alter the nitrate concentration, or when high nitrate fertilizers run off into the water. This happens in the gut. And so this is a vicious cycle. The high nitrate environment invites the growth of pathogenic bacteria, which then generate more inflammation and maintain this nitrate cycle. Now, there are a number of, so basically the foundation for reversing gut bacterial dysbiosis requires controlling intestinal inflammation. There are a number of natural products that I use for this purpose: quercetin, mastic gum from the Mediterranean food Pistachio lentiscus, curcumin from turmeric, omega-3 fats, magnolia bark, which is used a lot in Chinese medicine, and bovine serum immunoglobulins have all been shown in controlled studies to reverse intestinal inflammation, and I will choose from among those as part of the treatment. Because some of these also have antiviral effects, they do double duty. Quercetin has especially been studied for its antiviral effects and its anti-inflammatory effects in the gut, and there's several studies to that effect. Now, improving the gut microbiome. There is one study which is consistent with the results of many others, which showed that the gut microbiome at disease onset predicts the development of PASC. Previous studies had looked at people who had been hospitalized with COVID and looked at them after hospitalization and found very similar features to these features which were found to predispose to the development of long COVID. People who did not show these features had a much better outcome and recovered fully from COVID-19. First of all, there is a shift in bacterial concentration, and I've highlighted what I think are the three most important findings. There is a keystone bacterial species called Faecalibacterium prausnitzii, which is a major producer of a short-chain fatty acid called butyrate that has important anti-inflammatory effects in the GI tract and in the body as a whole. F. prausnitzii is diminished. There are higher levels of certain other bacteria that are associated with inflammation. In particular, a species called Ruminococcus navis. Now, you may not have heard of these. These are not available as probiotics, but they are very important, and the balance between them is very important in gut health. Bacteria that produce butyrate, their activity was inversely associated with the risk of developing long COVID. And F. prausnitzii is a major butyrate producer. So butyrate protects against long COVID. Loss of butyrate predisposes to long COVID. When we look at the physiology of butyrate, it becomes pretty clear that this is not just a coincidence. So step 3 is to try and restore the depleted bacteria and restrain the inflammatory bacteria. Once again, in a general way, bioflavonoids play a major role in doing that. Flavonoids I view as curators of the gut microbiome, and they encourage the growth of and activity of certain species and inhibits certain other species. And at the same time, we need these flavonoid dependent bacteria because they make the flavonoids more available to us. So butyrate is what is called a postbiotic. It is produced by bacteria in the gut. It's not produced by human cells. It has nutritional effects in the gut. About 80% of the energy that maintains the integrity of the lining of the large intestine comes from butyrate. The mitochondria in the large intestine depend on butyrate for their activity. It is produced by gut bacteria that ferment certain kinds of fibers and complex carbohydrates. Outside the GI tract, butyrate has some important effects. It has effects in the brain. This is a volatile short-chain fatty acid. It passes very readily through membranes. What's produced in the GI tract goes into the body. What circulates in the blood goes readily into the brain. One of the systemic effects of butyrate, it is an inhibitor of an enzyme called histone deacetylase. Histone deacetylase inhibits certain genes, and it has been shown that in the brain, the presence of butyrate activates some quiescent genes, in particular, a gene that's involved in producing a protein called brain-derived neurotrophic factor, BDNF, which is really important for recovery of neurons from damage and injury. It also activates various complexes on cells called G protein coupled receptors. Some of them are only activated by butyrate, and they have important effects in regulating immune responses and inflammation. So the steps to be taken toward reversing gut bacterial dysbiosis among people with long COVID. Number one, from a dietary perspective, the science would suggest a high fiber polyphenol rich diet that increases butyrate secretion production. It increases the growth of Faecalibacterium prausnitzii. I have some colleagues who prefer other types of diets. In looking at the science of what will correct the gut dysbiosis that follows COVID-19, a high fiber, plant-based, high polyphenol diet really is the one that meets the research criteria. And an adherence to a Mediterranean type diet was also shown to elevate fecal levels of Faecalibacterium prausnitzii. There are prebiotics that can be used, oligosaccharides, fructooligosaccharides, galactooligosaccharides that increase the growth of F. prausnitzii. Resveratrol is something that I've used for a different effect. There's a species of strongly inflammatory bacteria called Enterococcus faecalis that's associated with worse outcomes in people hospitalized with COVID-19. It is a major stimulus to the production of gamma interferon, which plays a role in the cytokines storm of acute COVID. Resveratrol inhibits the growth of E. faecalis, and also has effects in modulating the ability of bacteria to produce superantigens in the respiratory tract. There are two species of probiotics, strains that is, of probiotics, that have been shown to enhance the growth of F. prausnitzii in controlled human clinical trials. One of them is Bifidobacterium longum BB536. This was studied in children and adults with allergies, and at the same time that it had anti-inflammatory effects, it increased the growth of F. prausnitzii. And then there's a soil-derived organism called Bacillus coagulans, a particular strain, GB130, which was studied in elderly people and found to decrease inflammatory markers like C-reactive protein and increase the growth of F. prausnitzii. So I will often use those following the treatment with Bacillus subtilis. Now, this is a study that just came out on Ruminococcus navis. Now, you'll remember that higher levels of Ruminococcus navis was associated with an increased risk of long COVID. It turns out that Ruminococci, the whole genus that includes this, produce substances that may be toxic to the brain. In particular, there's a substance called isoamylamine, which, when studied in rats, was shown to quicken the pace of senescent cognitive decline. Stephanie will be talking about senescence later, and I think that the senescence, accelerated senescence in the brain, is part of the problem we're dealing with. Ruminococcus or Ruminococci and their metabolites may play a role there. Ruminococcus navis also converts tryptophan, an essential amino acid, to tryptamine, an amine that promotes migraine headaches and migraine phenomena. And migraine type phenomena play an important role in the physiology of neuropathic. So I'd like to move on to amino acids as they interact with the gut microbiome. We could spend a whole day talking about that. I'm just going to mention two of them. Tryptophan is converted to serotonin and to melatonin. Phenylalanine, another essential amino acid, is converted to tyrosine, which is an amino acid that is then deaminated to dopamine, decarboxylated to dopamine and norepinephrine, which are neurotransmitters. There are bacteria that produce these neurotransmitters, and there are bacteria that respond to their presence. What's especially important for ACE2, and I think Rachel will be talking about this more for COVID-19, is that ACE2, the cellular receptor for COVID-19, which is destroyed by interaction with the spike protein, has a special role in the gut. It is the chaperone that aids in the absorption of large neutral amino acids that include tryptophan and phenylalanine. And in fact, when there is impairment of ACE2 activity in the gut, tryptophan is the most effective. Okay, so one of the goals is to restore tryptophan serotonin balance. And we start by enhancing ACE2 activity. There are a number of natural products that will do that: vitamin D, curcumin, resveratrol, a variety of antioxidants, omega-3 fats. Fortunately, these have played other roles in antiviral roles and anti-inflammatory roles. Quercetin has complicated effects. It inhibits the breakdown, the oxidation of serotonin, but there was a study indicating it may reduce serotonin synthesis. So typically there's a point after about 2 or 3 weeks where I will pull serotonin out of the treatment to see how the person does without, to pull quercetin out of the treatment to see how the person does without quercetin. Tryptophan supplementation might be beneficial. There's an animal study that suggests that. I have not really been utilizing tryptophan in my treatment of patients with long COVID, but after reviewing this research, I will be looking into it. And there is a tryptophan metabolite called indole-3 propionic acid, which is produced by gut bacteria, in particular bacteria of the Clostridium species. That's a very complex species with some dangerous organisms and some very important one. Clostridium sporogenes is the one that's been identified as the major producer of this. This has anti-inflammatory effects. It has neurorestorative effects. And so there may be a role for tryptophan and balancing of gut flora to increase production of IPA, for healing the nervous system. Restoring the mucus layer and healing tight junctions are both important for restoring normal permeability in the intestinal tract after COVID-19. Many of the steps that we've already taken will do that. Bicalin, quercetin, the probiotic changes will all help with that. Finally, there's the question of, is there still toxicity in the infection, in the intestines? And is there something that needs to be altered? And this may even wind up being an earlier step in the process. One of the studies that was done, a couple of studies done with people hospitalized with COVID-19 showed fungal overgrowth in their stool persisting after discharge. And this fungal overgrowth was associated with fungal species that produce mycotoxins, as well as some pathogenic yeasts like Candida auris and Candida albicans. Now, there's a recent study in the US that found fungal cell wall beta glucans circulating in the blood of people with long COVID. And so there are a number of steps that should be taken to remove these fungi and their toxins and detoxify the intestines. Activated charcoal is one of them. I've been exploring the use of antifungal herbs as well. And then finally, enhancing the function of T effector memory cells, which are depleted in people with neuropathic. This may not occur across the full spectrum of patients with long COVID, but in a study from Northwestern University in Chicago, it was very clear that the function of T effector memory cells was one thing that all people with neuropathic had in common. Now, the gut microbiome has an important impact on the development of T effector memory cells. They butyrate, and fats and fatty acids in general enhance them. Fermented foods, lactobacilli in particular, enhance their development. And so, in addition to a high fiber, adequate fat diet, fermented foods like sauerkraut and kimchi are warranted. They are very dependent upon mitochondrial function. So at some point, we really need to have a symposium devoted to mitochondria in long COVID, because they show up everywhere. And this last slide is about Lactobacillus plantarum, which is found in fermented foods. There are also supplements. It grows on plants. There have been a couple of controlled studies, not in long COVID, but in other conditions in which Lactobacillus plantarum was really shown to impact immune function and recovery from infection in a very positive way. Thank you for your attention.
Thank you.
Thank you so much, Dr. Galland. I should appreciate the slides and information that you've managed to combine into these slides. And I know that you've looked into tens papers during this short period of time. And I should appreciate that you've designed such a beautiful presentation and the pack of knowledge for us to understand where basically neurological symptoms start. So gut health is really important. And as a representative for people who struggle to understand simple things like what type of probiotic they need to get. And I know you focused on Tundrex as a supplement to address antiviral properties that happen in long COVID. But if people can't get this particular type of probiotic, what is your view on going for a combination of different species, and the more species you get as a probiotic in a single solution is better, or people still need to focus on the key species, that keystone species you've mentioned?
Well, the keystone species that we want to build up, F. prausnitzii, which supports the growth of many other species, especially bifidobacteria. You can't get that as a supplement. So you have to build it up indirectly through diet, prebiotics. And then I showed slides on two probiotics that were shown to increase its growth. I don't think that the use of the Bacillus subtilis is essential. Also, it's a very short-term use. It's 2 or 3 weeks, no longer. We just want to get, what my goal is to try and get the spike protein out of the GI tract. Make sure that there's no replicating virus left. I can't guarantee that we're doing that, but that certainly is an immediate early goal. Then I want to try and build up a healthy gut microbiome, reduce the growth of ruminococcus, and a couple of things on that slide, by the way. Malvidin, a bioflavonoid found in berries, especially blueberries, inhibits the growth of ruminococcus navis. And again, a high fiber, plant-based, polyphenol-rich diet inhibits the growth of ruminococcus at the same time that it's building up Faecalibacterium prausnitzii. So I try to accomplish that mostly through diet, select probiotics, and Carlo, by the way, uses lactobacillus in his protocol. And so at some point, I will move over to lactobacillus species, usually the plantarum, as part of the attempt to enhance T memory cell activity.
I see. So overall, how to make sure you have quite good diversity of good bacteria, rather than you increase the rate of not good bacteria, which is bad and works against the good bacteria. So all these combinations with many species of bacteria, so it's not necessarily working good, as far as I understand.
There are 1,000 species of bacteria. So if you take a probiotic with 12 strains, most of which are going to be lactobacilli and bifidobacteria, which are really, the lactobacilli in particular, are really minor components of the normal gut microbiome. That doesn't mean they're not important, because number isn't the same as importance.
Yes.
But just taking more species rather than fewer, there's really no evidence that multi-species probiotics perform better.
Thank you.
So,
and I just want to add that this is a very individual thing. You need to work, you need to know your body. You need to pay attention to what's happening. I mean, I have patients who are taking multi-strain probiotics and they say, I really like this. This is the one that has helped me the most. I say, fine, stick with it. I mean, if you're getting that response to it, you're getting feedback from your body, pay attention to it.
Sure.
Dr. Galland, I got a couple of just questions for you. So when you talk about high fiber polyphenol diets, I think quite a number of abundant food contains, high fiber, those polyphenols. And there are a lot of nutritional supplements also being easily available in the market. So what do you suggest? People should take those supplements, will do the same as what if they take those natural fruits or whatever the sources they have in terms of the highest polyphenol contents and fiber?
Okay, so as a general principle, my belief is if you can do it with food, do it with food. However, a lot of people following COVID-19 will have GI symptoms.
Right.
And they may or may not be able to follow the kind of diet that I'm describing. So again, this is something you have to work out for yourself. I mean, the simple approach, especially for your people in Europe, and North America is to look to the so-called Mediterranean diet as a starting point. And there's just been so much written about it. In other parts of the world, look to traditional diets in the countries that you live in. I mean, the traditional Indian diet, the traditional Chinese diet, because traditional diets tend to meet those criteria. They tend to be plant-based and high in fiber and high in polyphenols.
Right.
And that's not an accident.
Sure. So, Dr. Galland, just continuing on that question. So, when we say, you get from your own diet, is there a particular concentration you're looking for all those polyphenols or high fiber diets from that? Is that what you're talking about, a specific concentration?
The total amount of polyphenols from diet, I mean, I don't think anybody plans their meals this way, but should be in the range of 4,000 milligrams a day.
Right.
And it is, for most people, it's hard to overdose on those. But there are some people who have certain genetic susceptibilities or genetic problems. They're not going to tolerate that, because one of the characteristics of bioflavonoids is they inhibit a lot of enzymes. And if you are genetically predisposed to, because the certain enzymes are weak, like the enzymes that methylate neurotransmitters, your tolerance for bioflavonoids may be much lower than someone else's.
Sure. So, Philip, you on mute. So, just a last question. Do you think that the antibiotic can be mixed with polyphenols, like amoxicillin?
Yes. Now there are yes, I think amoxicillin can be. There are certain antibiotics in which the polyphenols might actually interfere with the receptor binding, but not like the quinolone antibiotics, but not in this setting.
Right. Thank you. Thanks, Dr. Galland.
Yeah, I just wanted to ask a quick question from the audience here, Leo. This is from Rick. How long does it take for the virus to clear the GI tract in long COVID?
Without specific treatment, it may last at least a year. And there was a study that was done in people who were getting routine colonoscopies because of inflammatory bowel disease after having had COVID-19. And the people who had long COVID, but not the people who had fully recovered or the people who never had COVID, actually had spike protein in the cells. Not only spike proteins, there were nucleocapsid proteins in the cells lining the large intestine 12 months after having had COVID. And there was a hyperstimulation of certain types of immune cells in those specimens as well. It's actually certain types of T cells. That's a very complex process that we're going to need to really try and tease apart.
Wonderful. I'm going to ask us to move on to our next speaker now. So I'm going to shift over to Dr. Kamal, and I'm certain we'll have more questions later on, Leo.